in this case, what ratio are these observed numbers in? They set up an F1. What genotype does this F1 individual have? They don't use the slashes and semicolons like we do, but what's the genotype here? It's heterozygous at how many loci? How many genes? 2, so it's a dihybrid. So when you self a dihybrid, what's the expected phenotype ratio? That was the last chapter. 9 to 3 to 3 to 1. So the expected ratio is 9 to 3 to 3 to 1. And that's why I ask you, this is the actual observation. They set up a cross. They're using different traits than Mendel used. They're using a different plant. But they had a plant that had purple flowers and long pollen and a plant that had round pollen and red flowers. They make a dihybrid. They self the dihybrid. They expect to see 9 to 3 to 3 to 1. All the possible combinations of round and long pollen and red and purple flowers. What did they observe? What's the ratio? Is it 9 to 3 to 3 to 1, first of all? Is 296 to 19 to 27 to 85 anything like 9 to 3 to 3 to 1? No. So what is the ratio? How do we calculate the observed ratio? We see the expected ratio, that's from Mendel, 9 to 3 to 3 to 1. So what are the steps to calculating the observed ratio? So first you sum them up. So how many total observations do we have? 427. Okay. Then how do we figure out what the ratio is? Divide it by something. So how many squares are in this Punnett square? The 9 to 3 to 3 to 1 expected ratio. 16. So whatever this ratio is, it's going to total 16. 9 plus 3 plus 3 plus 1 equals 16. So we want to divide 427 by 16, and that tells us how many observations make up 1/16 of the total, which is approximately 27. So in a ratio, 27 individuals would be the 1. So you multiply that by 3, and that tells you, so instead of 9 to 3 to 3 to 1, we're going to figure this out in a little bit. That's how you calculate that ratio. So the observed ratio winds up being 11 to 1 to 1 to 3.
Dr. Ross?
Yep.
When we did that Excel sheet assignment that we did last chapter, you said that
Earlier, yeah.
Yeah, you said that we're not always going to get the expected ratio.
That's right.
Due to, so why can't this be explained by that?
And that's what we're going to do. So the question is, why is this not 9 3 to 3 to 1? So what do we need to do if we're going to ask that question? How can we address the question of, is this really a 9 to 3 to 3 to 1 ratio or not? Did I lose folks somewhere? Pardon? So chi-square test. So this is a perfect example of a place where we'd want to use, and that's that last question. Are the observed and the expected values different? So let's do a chi-square test. We'll do that at the end of class if there's time. But that's what you should do if you really want to answer this question. Is this a 9 to 3 to 3 to 1 ratio or not? So that's where we're heading today in part. So I'm almost done scoring the tests. There's one test left to be taken, so there's one left to grade, so I can't distribute your individual scores yet, but this is what the distribution of grades look like. It's very good for the whole class. The 2 Fs were the one person that hasn't taken it yet and one person that didn't show up, and I have not heard from that person. So nobody failed, few Ds, more Cs, more Bs, more As. Pretty happy. Okay. So we're going to talk about linkage. We're going to start talking about, these are the 7 different traits that Mendel used when he was crossing peas. So you can make lots of different combinations of all of these traits, and that's basically the question that we're here to answer today is, can we figure out how would you determine if you have an organism that you're working with in the laboratory and you want to make a particular combination of traits. Like in this case, let's say you wanted to make wrinkly green peas for some reason. The place you live, people love wrinkly peas that are green. I don't know. I want you to be able to answer that question of, can you actually do this with peas? Is it possible, using crosses, to combine those two traits together? Because it is the case that sometimes there are traits that you cannot make specific combinations of. So let's say that our goal is to make wrinkly green peas. Mendel did this, and this is what we talked about on the first slide. You set up a cross between a green round pea and a wrinkly yellow pea. You make F1s, you self the F1s or cross them together. And how many wrinkly green peas do you wind up getting out of all of this work that you put into months of crossing peas together? You get a 16th of your F2s, the one in the bottom here, are the trait that you want. And Mendel saw that this was the case regardless of which set of traits he was working with. The pinched pods versus inflated pods, pea shape, pea color, flower position, flower color. He always saw, when he set up those dihybrid crosses, that 9:3 to 3:1 phenotype ratio, just like here. So that was what Mendel saw. He was working with one species, but he was working with 7 different traits, and no matter which combination of traits he worked with, when you tried to make any combination of the two, he could do it, and it was always a 9:3 to 3:1 ratio. And then along came Bateson and Punnett, who put the data together we saw in the very first slide, where they didn't observe the 9:3 to 3:1 ratio. Purple flowers, red flowers, long pollen, round pollen. So what's different about Bateson and Punnett and what they did than what Mendel did? Why is there a difference? Why is there a discrepancy? What are the words we're using in this chapter? Linkage, recombination, crossing over. So Bateson and Punnett discovered something that had not been seen before, and it was something that we think Mendel didn't know about when he was working with his peas. So today we're going to talk about linkage versus independent assortment. That's a key distinction to make. Up till this point, with Mendel, we've been talking about the second law, independent assortment. We'll talk a little bit about nomenclature, and then most importantly, crossing over and how that disrupts the concept of linkage. Any questions to this point? Brief overview. So, I know they're not digital, but I brought my poker chips with me today. I've got 2 blue poker chips and I've got 2 red poker chips. So we're going to call red one gene and blue a second gene. We're dealing with 2 genes. How many alleles does each of these genes have?
1. Each would depend on the blue.
Well, they're shown up here. For this example, if I'm using a poker chip, it's like coin flipping when you do statistics, right? What's the probability that comes up heads? 50%. So this, how many alleles, how many different options do I have if I flip this coin? How many alleles are there? 2. Heads or tails. In this case, bicycle on one side or a spade on the other. Okay. So if I have a red gene and a blue gene from a mom, and a red gene and a blue gene from a dad, and I cross them together, I could make a dihybrid. I could get heads and tails, red and blue from mom, and I could get heads red, tails blue from dad. Somebody, let's see, who can help me with this? Who wants to help with this? A little coin flipping? Sir? Do you want to be the mom or the dad?
I'll be the mom.
Oh, that's the way. Okay. So before we do this, let's set up the Punnett Square. So this is the cross that we're about to do. My helper in the front and I. We're going to take a red coin that has a heads and a tails. So red H for heads, T for tails. So each of us has a red chip. It's got two sides, two possible alleles. So this is a homozygote or a heterozygote? Hetero. So this individual, this gentleman over here, who's going to be the mom in this cross, is a diploid, has one heads and one tails. Okay, for the red chip. And the same is true for the blue chip. Blue, there's a heads and there's a tails. And we're going to cross that to, so this is the dad side. We're going to cross that to mom. We both have the same chips. One red coin that has a heads and a tails, and a blue coin that has a heads and a tails. So what does the Punnett Square look like? How many squares does it need to have? Got two dihybrids, they're crossing together. So we're talking about two genes, dihybrid. How many rows, how many columns? It's 4 by 4. Typical dihybrid cross. So if we put red heads, we've got a gamete that has the red heads allele. You could have the blue heads allele, or red heads could go with blue tails. We're just predicting the outcomes of our flips when we flip our coins. What's the probability that he flips blue tails and red heads and I flip red tails and blue heads? So we've got his 4 possible outcomes. Red tails with blue heads and red tails, blue, what's left? Blue tails. And the exact same things, because we're crossing individuals with the same genotypes, go down this side. Okay, so once we're all set, we're going to flip these coins and we're going to tally where in this box they land. So we're going to put, let's see, this is going to be the dad genotypes on the x-axis and the mom there on the y. So who did I wind up being? Dad? Okay. So if I flip, I get bicycle. That's going to be tails.
Okay. So I put my blue too?
Yep.
It's the state.
Okay. So spade equals heads. Okay. So we got one heads, one tails. What about red? That time I got tails.
I got tails too.
So we got tails, tails for red, and for blue we got what? Heads, tails? Am I right?
Yeah.
Is that what we had? Okay, so where does that go in the Punnett Square? Hopefully you're writing this in. Where's this particular combination of genotypes? So I'm playing dad. I got blue heads and red tails. So we're in the third column. And then where do you land?
Red, yeah, red tails and, blue.
So this square. Okay. So we can make any combination, the two of us, flipping these coins, we could make any combination of traits we want. That's what Mendel told us, 9 to 3 to 3 to 1 ratio. We could have a cross that involves these two genes, we could hit any one of those squares. What's true about these coins? Which of these coins, which of Mendel's laws do these coins represent? How do they assort? Independently. What would this look like if we were talking about linkage? What would I have to do to my genes, my poker chips? Tape them together. So now they're physically linked, linkage. So now each of us, yeah, it's harder to flip these now. So the way that these are arranged is that we've got the red coin and the blue coin are arranged so that their heads are together on one side. And if you flip it over, you get blue tails with red tails. How does this affect what we find in our Punnett Square?
When you get one result, you're always sure to find the other. They both land together with a result, a certain result.
Right, so given that I just told you that heads go with heads and tails goes with tails, everybody close your eyes. It's like a trust experiment. Okay, sir, we actually, except for you, so flip your coin. No, it's not time to fall. So go ahead and flip and tell us what your red coin is. Everybody close your eyes. So the red coin is?
I got a head.
Got a spade. Okay, so what, everybody, time for the prediction. What's on the blue coin? If he got a spade when he flipped on the red, what did he see on the blue? Exactly, because they're linked. You can predict what's happening at the second gene if you know what's happening at the first gene, because they're physically associated with each other. So it takes the fun out of it. I got a red tail. Must have been a blue tail. I don't even have to look, because they're linked. So if the genes are linked, we're going to see what the Punnett square looks like. But this changes everything. This is why Bateson and Punnett didn't see a 9 to 3 to 3 to 1 ratio. It was because the genes that they were working with, the genes that controlled pollen shape and flower color, were linked. And that does something screwy to what Mendel would have predicted. So quickly, let's go over some nomenclature. So we're used to thinking about diploids. So I'll draw the same chromosome from dad and from mom for a diploid. So let's say this individual is homozygous for blue gene, homozygous for T, tails. Okay. Then we'll look at a separate chromosome pair. Again, one from dad, one from mom. This is where the red gene is going to be located. Also homozygous for tails. Are these genes linked or unlinked? If the genes are on separate chromosomes, are they linked or are they unlinked? Our coins are unlinked. So these genes are unlinked. What does it look like? What do I have to draw if I want to draw linked genes? They're physically, like these taped together coins, they're physically associated with each other, physically meaning that they're on the same piece of DNA. They're on the same chromosome. So I could draw a long chromosome. I could draw exactly the same genotype, homozygous for tails at the red gene, homozygous for tails at the blue gene. But now these two genes are like our poker chips, physically together. They're linked. They're on the same chromosome. How would we draw the genotypes of this chromosome on the left? How would we write it out? We've got the blue gene, tail allele. What comes next in our annotation? It's diploid, so there's, what's the other allele? We've got a blue tails here. What's on the other copy? Blue tails. What goes in between them? This is critical nomenclature. We're talking about the two alleles of the same gene. It's a slash. Okay. So we've got red tails over red tails on this other pair of chromosomes. What goes in between the two? This is where the semicolon comes in, because the semicolon indicates we're talking about separate chromosomes. Now the point here is that we change nomenclature when we deal with linked genes, because we're dealing on the right, as we drew it, it's just one pair of chromosomes. So what symbol is there not going to be in the genotype? If it's only one chromosome, we're not going to be writing any semicolons, because we're not talking about multiple chromosomes. We're just talking about one pair of chromosomes. So how do we write the genotype on the linked side? We still have red tails, but it's physically next to a blue tails. So we write out what's on one piece of DNA, the gene and the allele next to each other with nothing separating them. They're physically linked, so we write red tails and blue tails just like that. Now we're going to go work, now is there a slash? We're going to work on the other chromosome now and write out its genotype. So what do we put next? Now comes the slash. So we're going to stop talking about the first version of the chromosome. We're going to talk about the second version, which also has red tails, blue tails. So when you're talking about linked alleles, two versions of two different genes that are on the same chromosome, like here, you write them just next to each other. No spaces, no punctuation, anything in between them. And these are what we call, which is a term I've introduced before. Each of these is called a haplotype. Because that's the genotype you would find in a sperm or an egg, in a haploid gamete. So when you talk about the genotype of a chromatid, that's a haplotype. You put two haplotypes together with the slash in between, you make a genotype. So the two haplotypes here are identical, which is critical. There's no genetic difference between the two chromosomes on the right. Exact same haplotypes.
Dr.
Yeah.
You also got the same genotypes on the left.
It's exactly the same genotype as on the left. So the only difference between the left side and the right side is unlinked, linked. Unlinked on the left, linked is on the right. Where does independent assortment happen? Mendel's second law. Which side? Genes are going to be independently assorting, like the poker chips, on the left side, where the genes are on separate physical pieces of DNA. They're on separate chromosomes. So we've got independent assortment on the left side. What do we call what's going on the right side? Linkage. Or non-independent assortment, or dependent assortment, whatever you like.
Dr. Ross?
Yeah.
If on the independent assortment, let's say you had like big T, little t, heterozygous, then that mean for the right side you would also get the same genotype, just that they would be linked?
Right, so if you wanted to change You don't have to. It depends on the question or the system you're working with, but you could be heterozygous at that locus. And you could write exactly the same thing over here.
Oh, so you
You could be
still-
heterozygous for that gene in either case, linkage or unlinkage. In that case, one of these red t's would be a lowercase t, but this is just the nomenclature. Okay, so this is the most critical point. So if you're nodding off, now's the time to wake up, even just for half a minute. Punnett Square for linkage. How does the fact that the poker chips are taped together change what we draw in the Punnett Square?
It affects the haplotypes.
Right, so let's assume that we're doing the exact same cross we drew the 4x4 Punnett Square for earlier. So we've got a red heads, blue heads, these chromosomes we were just looking at. So this is a dihybrid genotype. It's just that the two genes are linked, crossed to itself. So it affects the number of haplotypes when genes are linked. How many different types of gametes will this parent produce? Let's write that on the y-axis of a Punnett square. Close your eyes. If I tell you that you see a meiotic product, a gamete with red heads, what has to go with it if it's linked? Blue heads. Do you get any red heads with blue tails? If they're linked, do we ever see the tape magically disappear and one of the coins flip over? Not in this example. So we've got two types of gametes. You either get red heads with blue heads or you get red tails with blue tails. So instead of having 4 rows, we have 2 with linked genes. How many columns are we going to have? The same parent, different sex, will produce the same gametes. Now we're ignoring recombination for the moment. This is the most simple version of dealing with linkage. Okay, so we're not going to fill this in, but the point is that this is why linkage affects what Mendel would have predicted. He would have expected to see a 9:3 to 3:1 ratio out of this cross. Instead, we're going to see something totally different. And what we see is that linkage gets disrupted. So occasionally in cells, the tape actually does get taken off. The alleles get separated and made into new combinations, crossing over recombination. So what's different about this, if you could explain in words, what's weird about the observations that Bateson and Punnett saw down here in the bottom? What's different? Where does the expected and the observed ratio really go just horribly wrong?
For the purple flowers, round pollen, and the red flowers and long pollen.
So for these two, we expected 3s and we got something like 1s. So we observed way fewer of those plants than we expected to see. What happened with the purple flowers long pollen and the red flowers round pollen, the one on the top and the bottom of the table? What was the pattern there? We observed more than or less than we expected. More. So here we observed fewer than we expected. But for the top and the bottom ones, we saw more than we expected. Where else do you see those phenotypes where we saw more than we expected to see? Where else on this screen are those particular combinations of phenotypes? Purple long and red round. Where do those exist up here? The parents. So for some reason, when genes are linked, we see more F2s that look like the grandparents, the P0 individuals, and we see fewer of what Mendel would have predicted would be recombinant types. So there are parental types, F2s, grandkids that look like their grandparents, and there are the recombinants, ones that would have new combinations, like maybe we're trying to make wrinkly green peas. We see many fewer than we would have expected of the new types. So we saw some recombinants, hold on. So we do see some of these recombinants, just not as many as we hoped for. Good.
So, I don't know if the genetics works the same way, but they say that if you were to cross self-fertilization, that you have genetic mutations, so that it wouldn't be possible. There's more likely chance that the children come off. So maybe could we hypothesize that because we see this difference, that maybe it's due to some sort of mutations and crossing over sections?
In this case, not mutations. So mutations isn't likely the answer to why we see fewer recombinants than we expect in this case. There are cases where that would be true, but generally speaking, no. Okay. So why do we see fewer recombinants than we expect? We have to go back to Mendel's traits again. He saw that 9 to 3 to 3 to 1 ratio every time he set up a cross between these traits. I want everybody to turn off any recording you're doing because I'm going to say something that's totally heretical again. And my colleagues will probably hate me for pointing this out. My genetics colleagues, anyway. So there's this idea in the genetics community that Mendel lied to us. But it's an interesting thing to consider, I think. And there are a pair of papers here that I've included in your slides, just the first paragraph of, for the aficionado, Mendel's celebrated paper from 1866 is studied repeatedly, much like an avid sports fan rewatches the World Series game or something like that. So I apologize on behalf of my genetics colleagues who are really, really into genetics, but it's true. Lots of people go back and look at Mendel's data again. What does this say? What did Mendel do? What do his data look like? It's generally agreed that his overall results are, pardon? They're good. They're too good. Did he fudge the data? So there have been published literature, people going back and looking at his original data and trying to use statistics against Mendel to prove that his data are too good. They're better than you would expect by chance. That his 9 to 3 to 3 to 1 ratios, maybe he fudged the data. Because they're too statistically perfect. They're too close to 9 to 3 to 3 to 1 ratios. So people have written whole papers and analyses of his data to try to figure out, did he lie? It's kind of like forensic statistics or something. So here's the question. Mendel used 7 different traits and all of the dihybrid crosses, trying to make all the combinations of all the different versions of the traits that he was working with. He always reported 9 to 3 to 3 to 1 ratios. What does that tell us about the genes? So if you see a 9 to 3 to 3 to 1 ratio, what does that tell us about Mendel's second law? Independent assortment. It's happening or it's not?
It's happening.
Yes. If you see a 9 to 3 to 3 to 1 ratio, that's what Mendel predicted from his second law. Independent assortment is happening if genes are independently assorting. They're on separate chromosomes. They're unlinked. So presumably, if we took a look using modern genetics techniques at peas, would we find that all of the genes that control all of the traits he was working with are unlinked? Are they all on separate chromosomes? So here's a genetic map of peas. Peas have seven chromosomes. The gene for flower color is found on chromosome 2. The gene for pod color and for height are found on the same chromosome, chromosome 3. Flower position, the gene for where on the stem the flower is located, is on the 4th chromosome. The 5th chromosome has seed shape, and the 7th chromosome has a gene that controls pod shape. So of his 7 traits, they're distributed across 6 of the 7 chromosomes in peas. Which are the 2 genes that are linked? Pod color and height. They're linked, they're on the same chromosome, but they're pretty far apart from each other on the same chromosome, and that's important. So this brings up an important point that's a fine distinction and one that I make that not all geneticists make, which is there's a binary discussion, or there's a binary decision. Either genes are linked or they're unlinked. If they're on separate chromosomes, they're unlinked. If they're on the same chromosome, we say that they're linked, like is written here. But they're kind of, we have to add a qualifier now. How close together physically are those genes? Are they really linked? Are they kind of distantly linked? They're linked, they're on the same chromosome. So I'm going to add an adjective. This is not real genetics terminology. This is just me trying to say that these genes are kind of far apart from each other on the same chromosome.
There's no threshold?
No, that's what I'm trying to say. There's no point, there's no formal terminology in genetics to describe whether genes are, there's no series of adjectives. Perfectly linked, somewhat linked, distantly linked.
Needs to be researched on.
Okay. So it seems like, what about this claim about Mendel lying or fudging the data? We're not going to do this, but I'm going to ask you the question anyway. What's the probability that by chance Mendel happened to work with 7 traits, and those 7 traits are distributed across 6 of the 7 chromosomes that pea plants have? Seemed like a pretty reasonable chance that he would pick 7 traits and that each of those traits is controlled by a gene on a separate chromosome? I see a few heads shaking. I generally agree. It seems really unlikely. We could do the statistics, but we won't. But it's unlikely, statistically, that he would have done this randomly. So what do you think Mendel actually did? Pardon?
He noticed the traits separately.
Maybe he studied more traits than we're learning about. So the textbook says these are the 7 traits he studied. Those are the 7 traits he reports in his papers. How many other traits do pea plants have? Lots. So it might be that he started working with 50 traits. Might be. I'm not saying anybody has proven this, but this is the conjecture. That he started working with 50 traits. And at the end of the day, he picked the 7 that met his prediction, that acted like they were unlinked. And those are the numbers that we have today, and that's how, and regardless of whether or not he lied, he still predicted laws of genetics that are true. Segregation of alleles and independent assortment, regardless of whether or not he fudged some data. But still, you have to wonder, can you trust the Austrian monk who's maybe picking and choosing data points to report to us? Those monks, I tell you. Okay. So what we learned from Bateson and Punnett and their crosses with the long pollen and the round pollen and the purple and the red flowers is that genes can become disrupted. Linkage can be disrupted. The tape can be taken off of the pair of poker chips, and you can make new combinations of alleles even if they're linked, even if they're on the same chromosome. And that's where crossing over comes in. What molecules are shown on the left here? It's a micrograph of chromosomes. How many chromosomes are there here? Or chromatids? Hint, hint. So if I draw this, this is kind of what it looks like. How many double strands? Each of these, sorry, let me be explicit. Each of those is a double-stranded double helix, each of those single lines. It's one double helix. So there are 4. If there are 4 chromatids all together lined up, when was this micrograph taken? Just after synthesis. So it had to be a diploid organism. So we went from 2 chromosomes to 4. They're all lined up, which means, what is this, mitosis or meiosis? If 4 sister chromatids are all lined up together, that's meiosis, and that's called a tetrad. This is a meiotic tetrad. So this is right before the first division of meiosis. And what's happening is, it looks like the chromosomes are physically breaking and switching parts with each other. And that's what crossing over is. In meiosis, meiosis 1, when those 4 sister chromatids are all lined up in a tetrad, the chromosomes physically break and switch parts with each other. Okay. So on this slide, I show, now we're back to talking about Bateson and Punnett, the P, purple or red gene, and the L, the long or the round pollen genes. So after synthesis, we start with a diploid F1 here. After synthesis, you've got that tetrad with big P's and big L's. And little p's and little l's. Okay. So just like every meiosis, you get 2 copies of every parent's chromosome right before you start making the divisions that make haploid gametes. 2 big p's, 2 big l's, 2 little p's, 2 little l's. Let's number the 4 sister chromatids. 1, 2, 3, 4, just to make the next step a little bit easier. And let me suggest that we're talking about, now we can start defining what we call genetic intervals. So regions of the chromosome. If we're working with 2 genes, there are 3 intervals. We'll call them A, B, and C. So there's the region above gene A, there's the region in between the 2 genes we're talking about, and there's the region underneath the second gene. So this is meiosis, right before meiosis 1, tetrad. Somebody pick a number between 1 and 4. 2. And tell me, thank you. Okay. So somebody else tell me a number out loud. Different number. 4. Okay. Student number 3. Say either A or B or C. A. Okay. So what happens during meiosis if sister chromatids 2 and 4 have a recombination event in interval A? What's going to happen? Nothing? That's pretty boring. So let's see. So if sister chromatids 2 and 4 break, and usually these recombination events are drawn as X's. So what's going to happen is this bit of chromosome 2 that I'm circling in red breaks just below that red circle and fuses to this part of chromatid 4. So if we redraw the new chromosomes, sister chromatids, after crossing over, you get what? Chromatid 2, as I've drawn it here, this part circled in red, would have a little p and a little l. As you go down from the top over here, little p, little l, down. So that's the new chromatid 2. What happens to chromatid 4? Circle what it does in blue. So if we draw the new version of chromatid 4 on the right, what's its haplotype? What combination of alleles does it have? In blue, right there. Does it have a big P or a little p? It's got a big P. It's got a big L. What about chromatids 1 and 3 that didn't do anything? There was no breaking, there was no crossing over. What does sister chromatid 1 look like? It's exactly the same. Big P, big L. Chromatid 3 looks exactly the same. There's a little p and a little L. So yes, I agree, that was a boring random choice of which sister chromatids could participate in recombination and which interval the recombination happens in. If we were going to do this again, what letter would you tell me we want to have recombination happen in for it to be more interesting? B. So if the recombination event happens, the breaking and the recombination of sister chromatids happens between the genes, now we start seeing haplotypes that we'd never seen before. New combinations of alleles, like big P's with little L's, or little p's with big L's. It's something worth playing around with. So to conclude, we need a few rules, and then I'm going to set you up with a couple things to work on for next time. What trait, what factor, what characteristic dictates whether or not two genes are going to have that recombination event happen between them? What would we do to make it more likely that a recombination event happens between two genes? Put them farther apart on the same chromosome. So physical distance between two genes is somehow related to whether or not that breaking and recombination will happen between them. Does every single tetrad do recombination in every meiosis? Yes or no? Will there always be a recombination event somewhere on the tetrad? Yes. How many recombination events or how many crossovers happen in one tetrad? The micrograph up here shows us, looks like 3. There are 3 spots here where the chromatids are crossing each other. We're going to assume for this class, for the sake of simple calculation, we're just going to assume that every tetrad has a single crossover happen. The math gets a lot more complicated the more crossovers you have. So for our class, that's how we're going to proceed. Now, the next 2 slides contain some questions I'd like you to think about, given this background. If there are 2 unlinked genes, how many of their chromosomes or how many of their gametes are going to be recombinant? So you'll need to draw out, here's what I would suggest doing, and this will be it. If we're talking about unlinked genes, we need to have a dihybrid. So let's say we've got this case for unlinkage. That's a tetrad at meiosis, that whole thing. There's a tetrad. The L gene is on a second tetrad, so they're unlinked. Do meiosis, do independent assortment, and then just answer the question. How many of the gametes are recombinant? How many are parental? So the recombinant types would be big P with little l and little p with big L. And the next slide asks essentially the same thing. If 2 genes are absolutely linked, and by this I mean, when I say absolutely linked, there's one of those adjectives, that the 2 genes are right next to each other on their chromosome. They're so close that they're touching each other. There may be even overlapping. That's what I mean by perfectly linked. 2 genes so close to each other on the same chromosome, they're practically indistinguishable from each other. So same question there. If meiosis happens and crossing over happens, how many recombination events are going to separate those 2 genes? How many parental type and how many recombinant gametes will you get? Okay? So we'll carry on with linkage and recombination mapping next class.